So here today we are beginning this course on good clinical laboratory practices as per ICMR guidelines 2021. So this course will be divided into 8 parts. Here in this course we are going to cover all the details of the ICMR guidelines related to the good clinical laboratory practices, and as per the recent NMC circular, it is mandatory for all the postgraduate to attend this good clinical laboratory practices course or have to undergo this good clinical laboratory practices training. Many of you might have undergone various trainings related to the laboratory practices.
Many of you must have undergone a lot of other trainings with respect to the clinical laboratory. You must have undergone ISO 15189 training, most of you people. So all these trainings are with respect to the international guidelines. Now, this particular course which we are here is with respect to the clinical laboratory practices as per the Indian scenario. So these guidelines were brought by ICMR in 2021. So in this first session, what are the things where we are going to cover is like introduction of the course and introduction of this ICMR guidelines. Then there are various definitions. So we'll see them in short about those definitions. And then we'll also see the scope of this document, ICMR document, where it is applicable. Then we will also see the different level of laboratories where this can be applicable, and then the next part will be the infrastructural requirement for these various levels of laboratories. So all these things will be covered in this first session. As all of you are knowing that there are 8 sessions of this program, so we are trying to cover complete guidelines from ICMR as per the good clinical laboratory practices. So first of all, coming to the introduction part, let me tell you that this is not the first edition. The first edition of GCLP guideline came in 2008. The first edition for good clinical laboratory practices guideline came in 2008. And this document is applicable to all type of laboratories which are involved in routine medical practices, and even for the research laboratories, whoever is handling the human samples, these guidelines are applicable to all those laboratories. These laboratories will help the clinical laboratories as well as the clinical researcher. Wherever this data is produced from number of researches and whatever research is being performed in the field, or whether it is performed in the clinical setting. So data which is produced from those laboratories, we want to make it reliable. So for that purpose, ICMR has introduced these guidelines, and which will also help in betterment of the healthcare services and health research in India. So coming to the overview now, as I have mentioned, this is the second edition of this document, and the first edition was in 2008 for the GCLP guidelines. Now, what are the things have changed during this year or in the second edition? So if you see the first part of the GCLP document in 2008, the clinical research laboratories were not included in those guidelines. So here in this new guideline 2021, clinical research laboratories are also included, and also there have been more emphasis on pre-examination and examination processes of the samples from the clinical research side and the testing laboratories side. So that is the new addition in this document of 2021. Now, these guidelines will help you to improve the safety and integrity of the test result. So whatever results we are producing in our laboratory, to make them more reliable, to make them more reproducible, or to make them more accurate, these guidelines is going to help you considering the quality standard, ethical conduct, and regulatory requirement. So basically, this document helps to promote the quality data, test data, which is produced from your laboratory, and it will also help in the why you have to rely on these clinical laboratory practices or good clinical practice. So whatever researches are conducted in the clinical setup, to make them more reliable, to make them more evidence-based, you need certain laboratory investigations. Now, if the data itself is not reliable, the outcomes of this research will not be appropriate. So to make them more appropriate and to make them more reliable, the ICMR has come with this GCLP guidelines. So here, good laboratory practices define a quality system concerned with the organizational processes and the conditions under which laboratory studies are planned, performed, monitored, recorded, archived, and reported. So basically, it covers all the aspects of a laboratory, that is starting from the planning of the laboratory studies, how they are performed, laboratory examinations, how they are performed, how the quality is monitored, how the functioning of the laboratory is monitored, how the data is recorded, how the evidences are recorded, and how they are archived. Archive means how they are stored for a long-term basis, so that whenever it is required in future, how they needs to be archived. So that will help for the future prospectus if it is required, and how these results are reported. So these all things are covered in this good clinical laboratory practices. Now this aims to establish a minimum criteria which should be followed in addition to the internationally accepted guidelines. As in the beginning itself I have mentioned that there are international guidelines available. ISO 15189 is one of the guidelines which is applicable to the medical laboratory, but we know in Indian scenario, it is not always possible to stick to those guidelines, or due to lack of resources, due to lack of facilities. These guidelines may not be applicable to the small laboratories or even for the government laboratories or laboratories which are working under research laboratories or on no profit laboratories. So to make them more customized, ICMR has come with their own guidelines. So these guidelines are basically the minimum criteria. Now, if you are not able to follow the international criteria, then what should be the alternative? So ICMR came with this minimum criteria that could be followed by the laboratories in India. So they call them as a good clinical laboratory practices, which are minimum criteria or minimum requirement which a laboratory should follow. Now, this was with respect to the introduction. Now coming to the definition. So there are various definitions. If you people have gone through this document of ICMR 2021, they have given several definitions. So all those definitions we are going to see in short. Most of you will be aware of these terminologies. If you are not aware, this is one of the opportunity to know about the meaning of all those terminologies. So first of all, we'll start with the analytical sensitivity. So what is in the analytical sensitivity? It is the ability to detect very low concentration of a given substance in a biological specimen. So what is the minimum concentration? We know many of the analytes are measured at very high concentration, but there are certain analytes which are present in a very trace quantity or at very low concentration. So that will be helpful to determine this analytical sensitivity. So it is the ability to detect very low concentration of a given substance in a biological specimen. Now, after analytical sensitivity, there is analytical specificity. So what is meant by specificity? So if you are going to measure one specific or one analyte, the other analyte should not come into the picture, or the other measure and should not come into the picture. So to avoid that, you should have a very high specificity. So that is analytical specificity. So it refers to the ability of an assay to measure particular organism or substance rather than others in that sample. So other samples, other analytes, other organisms should not interfere or should not come into the picture. That is the analytical specificity. Now next is the batch number. So as you will be seeing that all these definitions may not be technical which we are using in the laboratory, but these are related with the your laboratory practices. So what is batch number? So concentration, specificity, pH, impurities, etc. of the various components varies in different batches. Thus, information of batch number, date of manufacturing, and date of expiry of the reagents kits should be clearly mentioned on the label. So basically, batch number is nothing but the lot number or the batch number in which these reagents are prepared. So many a times there will be a minor variation with respect to the various batches or various lots of the particular reagent. So to address this issue, we should know which reagent was manufactured in which batch, what is the lot number, when it was manufactured, what was the date, what is the expiry of that reagents, or what is the expiry of the kit. So that is in your reagent bottle or under your reagent packages. Sometimes the expiry date you will not be able to find out, or you will not be able to see, or maybe due to storage condition it will not be visible. But if you have a particular batch number, if you have a particular lot number, from that you can find out what is the expiry of the reagent and what is the date of manufacturing of that reagent. So it is important to mention the batch number. Then next is the bias. So bias means basically the difference between the expectation of the test result and the measurement of result on the true value. So what is the true value? What is the exact value? What is the accurate value of the analyte and what is the estimated? So the difference between the expected value and the actual value is called as a bias. Next terminology is biological reference interval. So most of you will be aware what is the biological reference interval. So specified intervals of the distribution of values taken from a biological reference population. So what concentrations will be found in a particular population? What are the different levels will be found with respect to a particular analyte in the population you are serving? So that is a biological reference interval. Then next is the calibration. So basically calibration is the set of operations that establish under specified conditions the relationship between the values of the quantities indicated by a measuring instrument or measuring system or values represented by a material measure or a reference material and the corresponding value realized by the standard. So calibration is nothing but the standardization of your examination method with respect to a reference material. So you can see it should be with respect to a reference material. So there should be a appropriate reference material. Whenever you are standardizing any examination method, you have to standardize it with respect to a reference material. So that is called as a calibration. Next is the competence. It is the demonstrated ability to apply knowledge and skills. So nowadays there are more emphasis on competency of the staff, competency of the personnel working in the laboratory rather than their qualification. So basically competence is basically your ability, the ability of the personnel working in the laboratory to apply knowledge and skills to carry out a particular examination. Next is the critical interval. What is critical interval? It is the interval of examination results of an alert or critical test that indicate an immediate risk to the patient of injury or death. So any examination result which can pose immediate risk or immediate harm to the patient will be called as a critical interval or critical result. Next is the examination. So examination is basically a set of operations having the object of determining the value or characteristics of a property. So basically these are the operations or these are the step-by-step procedures which should be followed to determine the value or characteristics of a property. Of a property means of a measurand, of a analyte. So what are the methods? What are the different procedures you are following it? All those set of operations are collectively called as examinations. Next terminology is external quality assessment or proficiency testing. It refers to the system in which the performance of a laboratory is assessed periodically and retrospectively by and consider that your performance is monitored retrospectively and on periodic basis in comparison with the independent external agency or in comparison with the independent laboratory or a third party agency or maybe called as a laboratories, because there will be multiple laboratories where your performance will be evaluated, which will be your performance of your laboratory will be compared with various different other laboratories. So it is carried out by an independent external agencies to indicate any shortcomings in the laboratory performance through external quality assessment program or proficiency testing program. So basically it is a third party evaluation of the performance of your laboratory performance or laboratory quality assessment or proficiency testing program. Next is the internal quality control. It refers to the set of procedures undertaken by the laboratory personnel for the continuous and immediate monitoring of the laboratory work in order to decide whether the results are reliable enough to be released. Now, if you see a major difference here between the external quality assessment and the internal quality control, here it is, you can see what it mentions: continuous and immediate monitoring. continuous and immediate monitoring of the laboratory work. Whereas in external quality assessment, you can see what is the difference. It is a periodic and retrospectively. It is not a continuous, it is not a real-time process. It is a retrospective assessment of your laboratory's performance. Whereas internal quality control is a continuous and immediate monitoring. It is in a real time. We are going to monitor the performance of our laboratory and we are going to decide what results are generated. Are they reliable and are they can they be released to get the appropriate patient care? So that is the importance of internal quality control. Now here, this is one more terminology, investigator. Now where this investigator has come, from where this terminology has come, as I have mentioned, now these new guidelines are applicable even for the clinical research laboratories. So basically, investigators, this word comes from the good clinical practices. So where there are the researchers are the person who is responsible to conduct the clinical trials. That person is called as an investigator. There are principal investigator, co-investigators, which is defined by as per the ICMR guidelines. Now, next definition is a laboratory director. Laboratory director is a person or persons with responsibility for an authority over a laboratory. Now, this is the person who is having the complete responsibility and authority over a laboratory with respect to the all operations, supportive services, and management of the laboratory. So laboratory director is a person or person. So there can be more than one laboratory director who are responsible for the operations of the laboratory. Next definition is the measurement error. So to describe the difference between the laboratory measurement and the true value of the measurand. So what is the true value of the measurand or the actual value of the measurand, or what is the exact concentration of the measurand and whatever you have measured in the laboratory. So it may not be always accurate. There will be a slightly difference. There will be a slight difference. There will be slight uncertainties in the measurement. You may not be able to get the appropriate or exact concentration of a substance. So this difference is called as measurement error between your actual value and the values which is measured by your laboratory. Next is the measuring range. It is a range of concentration within the assay, is accurate and precise, and it should be documented. So measuring range means under or over which range minimum value and to the maximum concentration. The difference between this minimum concentration and the maximum concentration which your examination will be able to detect, or which your examination procedure will be able to measure. This range of concentration in which your results will be considered as accurate, reliable, and precise. This complete range will be called as the measuring range, and this complete measuring range spans from the minimum to the maximum concentration that can be applied. Next is outlier. What are these outliers? The observation in a sample so far separated in a value from the remainder as the suggest that it may be from a different population, or the result of an error is measurement. So basically outlier is nothing but when you are measuring certain values, and if some values are out of your average values, are out of your measuring range, those other values which falls beyond your measuring range or beyond your estimated values, those will be called as the outliers. So outliers means what was expected, or what was to be estimated, and whatever results which are generated, which are separated from the remainder values. So maybe you have hundreds of values with you, and 109 out of those 190 you have well in align, and which falls more or less together within a variation of 10%, but remaining 10% are away from these values or away from this group. So those 10 will be called as the outlier. Next is the post-examination processes, are also called as the post-analytical processes. These are the processes following the examinations, including review of results, retention, storage of the clinical materials, samples, disposal of the waste, and formatting and releasing, reporting and retention of the examination results. So once your results are generated, the remaining activities, after which, what are the activities you are going to perform? What are the activities you are going to perform? You are not going to release those results as it is. First of all, you have to review those results, whether they are accurate, whether they are reliable. You have to cross check with the clinical history. That means you have to review those results. You have to retain clinical specimens, or you have to discard clinical specimens. So whatever clinical material you have used for the examination activities. So those clinical material, those clinical specimens, you may retain or you may discard. So how they should be retained, how they should be stored. If you want to discard, how it should be discarded, and how the result should be written, or how the result should be transcribed in a specific format, and then how it should be reported. All this comes under post-examination processes or post-analytical processes. Next is the precision. So closeness of agreement between independent test measurement results obtained under stipulated conditions. So closeness of agreement. Closeness of agreement means how your results are close to each other. So when you repeat a particular test on the same sample within a period of short time, how they should be related with each other, or how the value should be appropriately resulted, how they are close to each other, how the results are close to each other. There should not be variation. Like one test you have performed, now it is 100 milligram percent, and after 1 hour, if you are performing it again, the value is 110 or 120. So there is no closeness of agreement. If you are repeating the same test again and again, you should be able to get the same value. You should be able to get the same result. That is a precision. So precision, basically this terminology used as a with respect to the qualitative estimation. If you are having a quantitative estimation, then the more appropriate terminology is a imprecision. So for numerical expressions of precision, the term imprecision is used, which is the dispersion of the results of measurement obtained under specified condition. So it is a dispersion of the results. If you are repeating the test on the same sample again and again, how the results will vary. That is a imprecision. That is a dispersion of the results of measurement. Next is the pre-examination processes and pre-analytical processes. So basically, these are the processes that start in a chronological order from the clinician request, include the examination request, preparation and identification of the patient, collection of the primary samples, transportation to and within the laboratory, and it ends with the analytical examination begins. So these are the basically all the activities which are covered before the actual examination starts. So maybe a patient comes to a laboratory or maybe goes to a clinician, then a examination request is raised. This request goes to the laboratory, billing is performed, then there is a request receiving is performed, then there is sample collection activities are there multiple. Then once the sample is collected, then there are various transportation activities are there. Samples are carried from one part of the your geographical location to another part of the geographical location. Many a times the samples are transported within the laboratory. So these transportation activities and the sample preparation activities. So before analyzing a samples or before performing the actual examinations, you have to prepare the samples. You have to take the appropriate samples, or you have to prepare the samples for your examination. So all these activities are covered under pre-examination processes. Next is the primary sample specimen. So primary sample specimen is basically a biological sample which you are going to withdraw from a patient. It may be in the form of blood, it may be in the form of any body fluid, it may be in the form of hair, tissues. You can see that it has been covered as a breath also, breath samples. So all those specimens, biological specimens which are collected from the patient, those will be called as the primary sample specimens. Next part is the quality control. So the process of monitoring and evaluating the performance of work by measuring that performance against established standards. So you have to monitor the processes. You have to evaluate the performance of your work against the established standard. So established standard means you will be having some criteria. So you have to evaluate your performance against those standards, against those activities. That is called as a quality control. After quality control, the next terminology is quality assurance. So what is the difference between quality control and quality assurance? So quality assurance is all planned and systematic actions necessary to provide adequate confidence that a service or product will satisfy given requirements for quality. So basically it doesn't only include the quality control activity, but going beyond, what are the other supportive activities which is required to produce a satisfactory product, or which is required to deliver a satisfactory services. All those activities are considered under a quality assurance. Quality assurance is the comprehensive term that refers to all the aspects of operations, starting from preparation of the patient to the sample collection, sample analysis, recording of the results, and its dispatch. So basically, quality assurance is a more broader term which applies to all the activities and all the operations of the laboratory. Next part is the quality management system. So again, we are going one step ahead. Quality control, quality assurance, and now quality management system. It includes all the activities of the overall management function that determines quality policy, objective, and implement them by means such as quality planning, quality control, quality assurance, and quality improvement within the system. So now we are going one step ahead. In quality control, we have seen that it is a real-time monitoring of your performance against set standard or set protocol, set criteria. In quality assurance, we have seen it is not only monitoring of your examination activities, but also the activities which are covered in pre-examination and post-examination, which helps to deliver a satisfactory services or satisfactory product. Whereas now quality management system goes one step beyond, and it also includes the other activities such as planning of the quality policy, quality objectives, implement them. Then it also includes quality planning, quality control, quality assurance, and quality improvement within the system. So it doesn't stop at only at the monitoring process. It also includes the quality improvement within the system. Next is the quality manual. So quality manual is the overall guiding document that defines your quality system through policies established by the laboratory. So basically it is a document where you are going to write down, where you are going to document what are the policies established by the laboratory, what are the quality systems. If you will get a complete definition of your quality system, you have to mention what are your policies, what will be your objective when you are working a laboratory in a laboratory to have a satisfactory product or to produce a satisfactory services. What are the different policies are required? All those should be documented in a quality manual. Next part is the raw data. So what is in the raw data? All original records, documentations, or verified copies of these generated by observations and activities during the conduct of the work. They are necessary for the constructions and evaluation of the result, reported results, and also called as source data. Now, raw data means when you are performing any examination and you are giving the result. So, what you have collected a patient sample, you have prepared the sample, you have performed the examination test, you got the result, you have evaluated it, you have reviewed the result, and you have reported. So what goes to the patient finally? A paper where the values or where the results are reported. But the result itself is not the all-in-one. You have certain background data or you have certain backup documents for it. So what it includes, you will have certain quality control measures, you may have performed certain calculations, you may have reviewed those results, you may have performed certain quality controls or calibration activities. So all this data is called as a raw data. So when we are resulting or when we are reporting a result, it is not just a simple single value. The single simple value comes with lot of processes, lot of observations, lot of evidences which you collect during the your examination processes. All those data, all those evidences, all those records which are behind your laboratory result are called as raw data or source data. Why it is called raw data or source data? Because From that only you have come to a final conclusion, or from that only you have come to a final result, or a final result which is reported to the patient. So it is called as a source data. Next part is the risk management. Risk management is an approach towards identifying and preventing the error from occurring, thereby avoiding harm to the patient. So risk is basically any potential harm or any potential uh, error. So in a risk management, you have to identify those errors, you have to identify those harms, and you have to rectify, you have to eliminate those harms. That is a risk management. Next is a safety data sheet. So safety data sheet, all chemicals, reagents are graded on a scale which poses a severe or potential life-threatening hazard. Thus, MSDS of all chemicals shall be available and should be handed to all users. So basically, in our laboratory, you might have seen we are using multiple chemicals, multiple reagents. We have acids, alkalis, other hazardous chemicals. So how to use them, how to handle them. So you should have complete knowledge of those chemicals, those reagents. You have acids, you have alkalis. Now there is a common protocol. Acids and alkalis should not be stored at a single place. Acids should be stored separately, alkalis should be stored separately, because if by accident there is some leakages or there is some breakages in the bottles of the acid as alkalis, they will react and it forms a very strong exothermic reaction. So to avoid that, they should be appropriately stored. Acid should be appropriately stored. The alkali should be appropriately stored. There are various chemicals which are carcinogenic. Those should be appropriately stored. Now, this all information comes under a safety data sheet. So you should maintain an appropriate data sheet for each and every chemicals and each and every reagent which is available in your laboratory. Those data sheets should also have information how to handle them, how to store them, what are the first aid precautions you should take when there is accidental exposure, how to discard these chemicals, what are the environmental hazards, how to act on exposure. All this information you have to mention in your safety data sheet. Next definition is sample. So one or more parts taken from a primary sample. So we may allocate one sample, one primary sample into several allocates, or we have a tissue histopathological specimen, we cut the specimen and we divide it into multiple small specimens. So that is these are all samples. So one or more parts which are taken directly from the primary samples. So primary sample, we have seen already, whatever specimen which is extracted from the patient, that is the primary sample. And what is sample? Basically, it is a part of your primary sample. Then next terminology is shift. Shift in the mean occur when an abrupt change is followed by 6 or more consecutive QC results that fall on one side of the mean, but typically within 95% range as if clustered around a new mean. On the 6th occasion, this is called a shift. So whenever there is a change in the mean, now what is meant by mean? Mean is the average values of your quality control result. So mean or average of your quality control result. If there is a shift means gradual change. If there is a gradual change in this mean, maybe on negative side or maybe on positive side. So for here, for example, they have mentioned that if 6 or more consecutive results, if 6 or more consecutive QC results are going only on lower side or on higher side, then the 6th result will be called as a shift. So this 6th result will be considered as a that there is a change in the shift or change in the mean. So whenever 6 or more consecutive QC results fall on only on the one side, the 6th result will be called as shifted from the mean. Next is the stability and shelf life. The shelf life and stability data of all the reagents and kits should be documented in agreement with the manufacturer's specification. So what is the shelf life? What is the span life? What is the open reagent life? What is the open reagent stability? All this data should be collected from the manufacturer. So when you receive a chemical or reagent, you will be having manufacturing date, and you will having expiry date. So this expiry date is basically a shelf life or expiry of the or the span life of the reagent. But it may change if you once you open the reagent bottle or once you decap the reagent bottle or chemical, the span life may change. That is called as the stability of the reagent. That is called the stability of the open reagent. Next principle, a definition is sponsor. What is sponsor? An individual, company, institution, or organization that takes responsibility for the initiation, management, and financing of a clinical trial. So basically, when and in the beginning itself, I have mentioned that these guidelines are applicable now to the clinical research laboratories also. So a lot of clinical trials are conducted there. So those clinical trials are initiated or stated by sponsors. Now, these sponsors may be from the governmental organizations or there will be some corporate private pharmaceutical industries also. Those who are sponsoring these trials, those who are sponsoring this research, they initiate, they manage, and they provide a financial support. They are called as sponsors. Next is standard operating procedure, or it is called as SOP. SOP contains step-by-step written instructions for each procedure performed in the laboratory. These instructions are essential to ensure that all the procedures are performed consistently by everyone in the laboratory. Now, we see why SOP should be available or why we have to prepare the SOPs so that whatever procedures you are performing, they should be consistently performed by everyone in the laboratory in the same manner. So if there are 10 persons working in the laboratory, all 10 should perform the examination in the same pattern, in the same manner. So for that purpose, there should be a proper instruction. So this set of instructions is called as standard operating procedures or SOPs. Then what is trends? Trends occur when values gradually but continually move in one direction over 6 or more analytical runs. So in shift we have seen there was also the values are shifting towards the one side. So there also we have taken a number, standard number, 6 values or more than 6 values. If they are falling only on one side, it will be called as trends. Trends may display values across the mean or the may occur only on one side of the mean. So trends can occur on any one side or even across the mean. So it may start from higher side to lower side, or it may be on only on the lower side, or it may be on the higher side. So the trends may cross the mean also, or it may be on lower side or only one side of the mean. So here also the sixth occasion will be determined as the trend or and the results are detected. So if you are getting such a trend in your quality control value, then the sixth value, if it falls again on the same side or it shows a particular trend on higher side or lower side or across the mean, then the result must be rejected. Then next definition is trial protocol. The overall study protocol approved by the sponsor, which describes the entire activities which make up the study. So protocols means again a step-by-step procedure. So trial protocol means a clinical trial protocol. When we conduct a clinical research, these are the clinical trial protocols. These protocols are given by the sponsors because as you have seen, they have initiated the study, they are going to manage the study, and they are going to financially support the study. So how to conduct the study is as per their protocol. So these are the trial protocol as per which we have to conduct the each study or each clinical trial. Next is the uncertainty of measurement. Is the expression used to represent the unavoidable error. It is the foremost responsibility of the laboratory to evaluate the reliability and quantify the unavoidable laboratory error for every analyte. Now we have already seen it is not possible to get the same exact actual result if you keep on repeating the test. So there is certain variation due to unavoidable error. There are certain errors or there are certain variations in your analytical process. There is certain uncertainties. So this uncertainty of your measurement, because as I mentioned, you may not get the same result. So if you are estimating a glucose value of one patient, So if you repeat the same test 10 times, 20 times, maybe first occasion you get the value of 90, next you are getting 91, again you got 90, again you got 90.5, 91.5, 92, 89. So there is a variation in your result. So what causes this variation? So there are certain unavoidable errors, or there are certain unavoidable factors. So these errors or these factors contribute to the uncertainty, or this gives rise to the variations in your measurement. That is called as uncertainty of measurement. Next part is the validation definition. It is the process of confirmation through the provision of objective evidence that the requirement for a specific intended use or application has been fulfilled. So now you have to see, we have two definitions here. I will club this both the definition together, validation and verification. So what is validation? It is the process of confirmation through the provision of objective evidences that the requirement for a specified intended use or application has been fulfilled. So here you have to confirm that whatever is the objective of your examination method, or what is the objective of your examinations, what is specific intended use, what is application, whether you are able to, whether your test method is able to satisfy the requirement, whether it is able to specifically satisfy the intended use or application. So if you have a test method and you have to see that this test method is used for this particular purpose, so you have to confirm, yes, this method can be used for this particular purpose. So that is called as validation. What is verification? It refers to the laboratory's confirmation of the performance of the equipment based on given specifications or requirement. It also confirms that instrument is working for its intended purpose. Now, once you have confirmed the application and intended use in the validation, what you have to perform? You have to perform the verification that whatever application or specification has been mentioned, whether it is able to actually reproduce, whether it is actually able to perform as per the intended purpose. So that is a verification. So here I'll give you a simple example. If you are developing a new test or new method, you have to perform the validation. If the test method is already validated, then you have to confirm whatever claims have been made by the manufacturer or whatever claims have been made by the examination developer. You are able to produce all the claims in your laboratory. So that is a verification. So verification needs to be performed by the laboratory. Validation is performed at the time of development of a new method or whenever you are going to modify the examination method. So these were some of the definitions which we have seen, or which we will be all these sessions. Apart from that, if there are any terminologies we come across in these sessions, and if you are not going to understand or if you are not able to understand, you can just put into the box in Q&A so that we can solve those queries or we can take those terminologies also. Now, once after that, we will see what is the scope of this document, to what kind of specialties, what kind of different specialties this document is applicable. So this document is applicable for the microbiology and infectious disease serology laboratories. It is applicable to hematology and blood banking, molecular biology and molecular pathology. It is applicable to clinical pathology, clinical biochemistry, routine and special assays or special techniques such as TDM and immunoassays. It is also applicable to the histopathology and cytopathology laboratories and genetics laboratories. So this is the scope of this document. To all these specialties and all these areas, this document will be applicable. Now, the next part is the levels of laboratories. What are the different levels? What are the different tier system, or what is the tier system which we follow in our India for the different kinds of laboratories? Now, as we have already seen that this is as per the ICMR guidelines. So this ICMR guidelines is specifically in relation with the public health laboratories. This is in related to the public health laboratories. So in Indian public health, we generally follow a three-tier public health system: primary level, secondary level, and tertiary level. So we have three different, or we have three-tier system in our Indian public health. Besides this primary, secondary, and tertiary level, there are various reference laboratories, research laboratories, and specific disease laboratories. You might have seen virology laboratory, TB laboratory, district TB laboratory, TB research laboratory. You will have HIV laboratory, HIV testing laboratory. So these are specific disease laboratories. So these are also a part of Indian public health system. So levels of public health basically at grassroots level. If you are aware of this system, we have at grassroots level, sub-center is there. Primary health level, we have primary health center. The secondary levels, we have community health center and sub-district hospitals. The tertiary levels are the district hospitals and medical colleges. As I have insisting that this is a public health laboratory structure in our India. This is related to the levels of public health care in India. So we have a three-tier system: primary level, secondary level, and tertiary level. And at grassroots level, we have a sub-center. So the patients, the first point of contact in remote locations are sub-centers. Then we have primary health centers where primary health care, primary preventive and curative disease services are provided. Then we have referral centers. These secondary centers are called as the referral centers. So those conditions which are not able to manage at primary health center or the facilities is lacking at primary health center, those patients are then the district hospitals and medical college. So these are basically the tertiary levels in public healthcare of the Indian scenario. So this is a healthcare system which we see in our India, public sector. So now the laboratories are also classified as per that public health sector. So first level of laboratory, primary laboratory, which is available at primary healthcare center. What is the capacity and infrastructural requirement? It should be appropriate. There should be appropriate staff. The clinical laboratory services should include essential laboratory services, PHC 8, ICMR, NEDL. Now, what is essential diagnostic laboratory services? So, this is a 2019 document. If anybody has come across this document, they can find out this is a document which was there or which was published by the Indian government in 2019. It classifies the test which should be available at primary health center, test which should be available at secondary or referral centers, and test which should be available at district hospitals or medical college level. So the list has been published by the government. So as per that, the facilities should be available in the primary health center. Then there should be appropriate quality assurance activities in this primary health level. There should be appropriate internal quality control and with possible inter-laboratory comparison. So again, the public health, they have their own structure to perform this inter-laboratory comparison. So how it works, I will give you one small example because this is related with the public health setting, and I'm not sure how many of you are aware how these laboratories in the public health sector works as far as this inter-laboratory comparison programs are available. So here what you have to do, you have to take if I am carrying out malaria test at my primary health center. So I have to take 5 positive slides and 5 to my referral center. For me, referral center may be a sub-district hospital or a district hospital, whichever is nearby my primary health center. So I have to send this sample and I have to compare my result with those laboratories. Similarly, for TB, sputum, or for sputum examinations, again, you have to take those slides, you have to perform a comparison with your referral laboratories or with your referral center, maybe a sub-district hospital or district hospital. So that is how a quality assurance program works in a primary health center. Next level is the secondary, where we have seen it comes under sub-district hospitals or referral community health centers. Here, the capacity and infrastructure requirement also, as per the appropriate staff range, you should have a designated area for clinical laboratory, including sample collection and storage. So here, you should have appropriate designated area for sample collection and processing of those samples. Again, here also you will have essential laboratory services and a list of essential diagnostic laboratory investigations which should be available at sub-district level. So this list is again published in 2019, and here also you should have appropriate IQC results and EQUAS monitoring activities. Then there is a third layer or tertiary level. Here the capacity and structure includes appropriate staff strength, easy access to OPD and IPD. There should be adequate layout, essential logical flow for sample receiving and sample disposal. There should be a separate demarcated area for sample collection, sample processing in hematology, biochemistry, clinical pathology, and reporting. There should be a services should be available as per the ICMR guidelines NEDL 2019. And again, there should be a quality assurance activities, which now here it doesn't not only includes with your IQC and EQUAS simply. It also includes standard operating procedures, appropriate training of the staff, and regular validation of the reports on regular basis. So all these laboratories also should follow appropriate biomedical and waste management guidelines, whichever is applicable to your specific geographical area. Apart from that, as I have mentioned, there are specific research laboratories, reference laboratories, and specific disease laboratories which are available at the level of medical colleges, or these are the standalone laboratories. As I have mentioned, specific research laboratories are available, and there should be those laboratories should maintain high quality of standards, so that these laboratories act as a reference center for all these small laboratories. They act as a reference laboratories for all the small laboratories at primary, secondary, and tertiary level. So there are certain state and national laboratories, reference laboratories are available, which provides you assistance on scientific and technical. They provide training services, counseling services, expert advices are available. This you might have seen during the period of COVID, that there were certain reference laboratories, national laboratories, which were providing the quality assurance program, which were helping you to develop the infrastructure. They have given the training, they have given the appropriate resources, they have provided the appropriate quality control materials, external quality assessment schemes they have provided during the COVID period. So this is the responsibility of all these research laboratories and specific disease laboratories, which is again a part of public health laboratory or public health system in India. Then there are certain collection centers. At grassroots level, we have seen there are sub centers where no processing or where no testing is performed, but they simply collect the samples and then they are sending it to other laboratories for the processing or for getting the results. So there are certain collection centers available. What are the infrastructural requirements are available? So here are certain basic points are given. Infrastructure of the laboratory varies with the requirement of the clinical research, should meet the regulatory and international standard. So the requirement should meet the regulatory and international standard. What type of infrastructure is required? How much space is required? What are the specific segregation is required? It should be as per the regulatory standards and international standards. Infrastructure of the laboratory directly impact the quality of the research data and efficiency and safety. So if you do not maintain a appropriate infrastructure, then there are chances of contamination, there are chances of invalidate results. So you should have appropriate infrastructure for working in the laboratory. The management of the laboratory is directly responsible for providing all the adequate source resources to the staff to ensure efficient working of the laboratory. So in a good clinical laboratory practices, there is something called a good laboratory design. So what should it include? There should be signage. This signage should be outside of the laboratory and inside of the laboratory. It should mention the name of the laboratory, registration number of the laboratory, what are the services offered by the laboratory, working hours of the laboratory, contact number, when the test results can be expected, that is turnaround time, it should be available. Then other signages should include health and safety regulations, your fire exit signs should be there, other chemical and biohazard symbols should be appropriately mentioned at various places in your infrastructural. Then there should be appropriate emergency and disaster management plans for fire, chemical spillage, and lab accident. There should be uninterrupted power supply. Now, this is very important requirement, that there should be an appropriate uninterrupted power supply in your laboratory. It may be in the form of generators. You should have appropriate backups, or you should have UPS or inline, offline UPS should be available in a laboratory. Laboratory grade water for examination purpose should be available. So you should have appropriate laboratory grade water, which can be available via distilled water plant, or you should have appropriate facility to procure this distilled water. it should be appropriately ventilated. There should be appropriate environmental control in the form of temperature, humidity, magnetic field, vibrations, lighting arrangement. There should be appropriate communication facilities with the referral center. So if some test is not available in the laboratory, then you should be able to appropriately communicate with the referral center. There should be arrangement for transportation of the samples. Lab areas containing carcinogens, radioisotope, biohazard lasers should be properly marked with the appropriate warning signs. There should be appropriate segregation of the reception area, waiting area, areas where cross-contamination can happen, such as microbiological laboratories, your molecular laboratories, where their contamination can cause or invalidate the result. It should be appropriately segregated. And whenever possible, you should have appropriate segregation, and areas should be available for space of handling for radioactive materials. If you are doing certain nuclear medicine test, or if you have appropriate, you should have appropriate waste disposal facilities, biomedical waste facilities for cleaning of the glassware, sterilization, disinfection should be available. There should be separate areas for meeting, training, administrative work. There should be separate areas for eating and storing of food, drink. There should be appropriate facilities for toilet for the staff. There should be appropriate eye wash facility, body wash facilities. There should be possible, if possible, there should be areas for meeting. A small library should be available whenever it is possible. There should be appropriate infrastructural facilities for storage of food items, storage of reagents, storage of chemicals, storage of your documents, records, slide storage, specimen storage area should be available as and when required as per the requirement of your laboratory. And then finally, there should be a restricted area. So there should be appropriate access control. Not everybody should enter in all the areas. If it is relevant to your work, you should enter it. If it is not relevant to work areas, you should not able those areas. So there should be a restricted access wherever the reagents, equipment are stored, wherever the examination activities are performed, wherever the chemicals are stored. You should have appropriate restricted access, or the access control should be available. Apart from that, additional infrastructural facilities may be added for special tasks as and when needed. So maybe if some laboratories are performing some highly specialized tests, or some very highly superior or highly high-end tests, then whatever infrastructural requirement, those should be followed as per the requirement of those specific facilities. So it is very essential and important to have infrastructural facilities to have a quality report and a quality data which is generated from the laboratory. So with this, we are concluding this first session of this program.